Hello. This video will demonstrate the proficiency in preparing of blood samples and biopsy samples to transport to an outside lab. First of all, it is very important to know the requirements of the lab in which you are sending your specimens to. So make sure the research is done before the blood is obtained from the patient, so the patient does not have to be stuck twice. We all want to avoid that. So let's say, for an example, we're going to use a mock patient named Amber, canine, and send her results to an outside lab for coagulation tests such as PT, PTT, and fibrinogen. It is very important for any coagulation test to be in the correct tube, and this tube is a blue top tube with an additive sodium citrate in it for the anticoagulant. Very important for coagulation tests that the ratio, this is a blue top tube, the ratio of blood to anticoagulant has to be sufficient. And there is a fill line on every blue top tube that you must fill with blood. There's no exceptions, because if you don't, the results will be erroneous or the test will be rejected by the lab. So you have to fill up this tube. So the next step would be to spin the blood down in the centrifuge, such as that one or that one. And then you would spin it down, and you want to separate out your blood from your plasma. Very important with coagulation tests, because we have to separate those cells from the plasma. Then we're going to take that plasma, which is the watery part of the blood, and we're going to put it into an aliquot tube. We're going to take that aliquot tube and we're going to label it properly with the owner's name, the dog's name, the date, what is in that tube. For instance, sodium citrated plasma. We want to know that at a reference lab. And so all that has to be on there. When that is done, we want to then take our plasma and we want to freeze it. One thing about coagulation tests is that the proteins rapidly degrade, and so if it's not going to be traveled to their outside lab in an appropriate amount of time, we must freeze our sample. And then when we transport our sample with the courier, that too must be transported with a frozen pack. Very important. Also, when you do your research in your book, it will tell you that, it should tell you all these directions on what is required for the sample collection and the transport mechanism. How do you transport it? Is refrigeration just okay with a cooler, or does it have to be frozen, like with dry ice? Something like that. Or room temperature. Some tests require that they be stored and traveled at room temperature. All this stuff should be indicated in the lab in which you're going to send your samples to. All these requirements should be there for you as the tech sending this stuff off to have this available to you. Now, if we needed to have serum, if a test required serum, we would use a red top tube. There's no anticoagulant in here, and this particular tube has gel in it. So I would obtain blood from a patient, and then I would let it sit for 30 minutes in an upright position so that it would clot. After that, I would spin it down in one of the centrifuges. And this particular tube, well, this gel right here would separate my serum from my cells, and that would be okay because I have a separator that I could send it like that to an outside lab. That would be okay. Or I could aliquot it into another tube, indicating on that tube that there is serum in that tube, also with the label requirements as I previously mentioned. Now, if I'm doing a CBC, for instance, I would need a purple top tube. This is whole blood, so no centrifuges involved. We would want to take the sample from the patient and mix it a few times. And when that is by refrigeration, stored and transported. And then we have another top tube, which is a green top tube, such as this one. And there's heparin in here, and that would be heparinized plasma in here. Okay, so there's an anticoagulant of heparin in here. Same thing, draw the blood, spin it down. This one happens to also have a gel top or a gel separator in there. So not only does it have heparin, but it also has a gel separator. Very nice. So we'll separate your cells from your plasma, your heparinized plasma, after it's spun in the centrifuge, and you can transport it like that. Okay, now if we have a microbiology sample, very important, we use a sterile swab. This is for like wound cultures, eye cultures, anything you want to culture must be in a sterile swab, and then you would take your sample sterilely, and then you would put it back in the little case because all of this is sterile, and you would label your tube correctly. Okay, so all that is for our lab work. Now, like for blood and for microbiology. Now, if you have any tissue samples that need to be sent to the reference lab for pathology, you would want to send that piece of tissue in a small vial of formalin. Formalin is a carcinogenic, they say, so it is very important that you be wearing gloves. You don't want to get this on your hands, and but it is a tissue preservative. So we would take that tissue that the doctor would want to be analyzed by the pathologist, and we would put it into this vial. Very important, we would label this vial. Same thing, patient name, owner's name, the date, the source, the doctor, all that, and you want to make sure you make sure the top is tight. And when you bag that container of formalin, you would like to double bag it just in case there is an accident. And in transport, you don't want formalin getting out there exposed in your courier or such. So with that said, I hope I've covered all the areas of specimen processing and transporting to the outside lab for outside testing. And also with those samples, you want to make sure you do sign or fill out a lab requisition for that laboratory. And on there is self-explanatory. You would fill out the name, owner's name, date, source, and all that information. And that will travel with your specimens. So I hope I reached all the points, the key points needed for proper specimen processing and tissue processing. And thank you. Hello. I have here in front of me a Sysmix 2000, which is a hematology analyzer. Now this is not the common analyzer you would find in a small animal facility, but larger animal facilities like the University of Georgia Veterinarian Hospital right down the street, or reference laboratories will have analyzers like this. With this kind of laboratory instrument, it is vital that the integrity of the results be checked every 8 hours. So how do we do that? We run our quality control samples, just like we would run our patient samples. Quality control samples are known values that must be obtained each time they are ran. So let's get started and run them now. We have a low and a medium and a high, and we're going to put them in the sample rack just as we would a patient. And I'm going to put it on the analyzer. So while that's running, let's talk about other stuff. I'm going to show you the QC chart that we review every time we run the QC. So let's go here to QC, and here we have our chart. Now, for instance, the last time the QC was ran, I would go to control value number 1, and I will double click on it, and I can see my chart. And here we have the QC chart. Now, I want to make sure my results are maintaining on the mean, which is the line. I don't want to go 2 standard deviations in any direction because that would be bad. So I want to make sure my results are going up and down the mean, like here, with the mean as the average of our known values. And that would indicate that this machine is in optimized condition, and that is good. Now, if we were trending 10 directions in any direction above the mean or below the mean, we know we would have a problem. We would have to address it, such as calibrating the machine, or open up new vials of new QC, or cleaning up the lines on the instrument. So, also maintenance on this analyzer, we check our reagent supply. So we would check it by hitting the reagent supply button, and here tells us numbers, and it indicates how many tests are left for these packs that are involved. And we're good here for now. So we don't need to change anything out. But should we have to change it out, it's pretty simple. We just press on it, and we would go ahead and change it out and then prime the lines. So not a problem, very easy. Also, every 24 hours we run a bleach sample through the instrument, just like we would a patient, right? Into this little tube here is bleach, and we run it through the instrument, and we will bleach out the lines and clean the machine. So pretty simple. Weekly we bring the whole analyzer down to rest it and bring it back up. And overall, that is pretty much easy. For this CBC analyzer, it's a fantastic instrument. It runs CBC, reticks, and body fluids with some limitation on that. So I hope I've covered everything on every 8 hours, and our reagent supply and our bleach, running our bleach through the lines. Hello. In this video, I will demonstrate an ELISA method. I'm going to use an IODEX 4DX Plus test kit, which is right here. And in this test kit, we're going to test our animal for antibodies against canine atoplasmosis, canine ehrlichiosis, Lyme disease, and the antigen for heartworm disease. They are all vector-borne diseases. The atoplasmosis, ehrlichiosis, and Lyme disease are all delivered to the animal by specific ticks, and the heartworm disease is delivered to the animal by the mosquito. This ELISA IODEX 4DX Plus test kit is a fantastic rapid test that can be done right in the vet clinic. The ELISA method is comparable to reference laboratory test methods. In contrast to the ELISA method test kit is the lateral flow test kits. So how does the ELISA test technology work? First, the sample of EDTA or heparinized whole blood, serum, or plasma is mixed with a conjugate to a sample tube. That tube is then mixed well by inversion and applied to the sample well area. The sample then has a bidirectional flow, giving it 2 chances for the sample to bind to antibodies. This greatly improves sensitivity. The next step is an integrated wash step that removes any debris from the read window, which is great. The last step is the amplification, where even low-level antibodies can be detected. So let's get started. So we have let this come to room temperature for 30 minutes because this is stored in the refrigerator, so it has to equivalent to room temperature, and that takes about 30 minutes. So what our supplies are is the test kit, our conjugate, our EDTA whole blood, and a test vial with some pipettes. So we're going to, and a timer. We want to set our timer. So we're going to open up our test. And there's a little notch on the little container. Okay, so this is the test, the SNAP test, okay? It's by IODEX. Okay, and that's our sample well. And there are the impregnated test well in there, okay? All right, so let's see. So, to our test vial, we're going to add 3 drops of whole blood. So let me mix that first. Okay, so I'm going to add 3 drops. 1, 2, 3. Then I'm going to add 4 drops of conjugate. 1, 2, 3, 4. Okay. Then I'm going to recap my test vial, and I'm going to invert it 3 to 5 times. So 1, 2, 3, 4, 5. I want to give it a good mix. I really want to give it a nice mixture without shaking. I don't want to shake it. Okay, so I want to put my kit on a horizontal surface, which it's on. And then I'm going to take all of my sample and conjugate mixture, and I'm going to apply it to the sample well right here. Okay? So I'm just going to use this pipette that I used the blood from, and I'm going to apply it all right into that sample well. Now, it's starting to flow up through the reed window, and why it's doing that, when the color first appears in the activation circle, we're going to push this activator here, and we're going to push it all the way down until it is flush with the device body. Right there. Okay, so that activated this test kit to start working. So in 8 minutes, we're going to come back and we're going to read that and see, hopefully her dog has no antibodies to those diseases mentioned, or the heartworm antigen, and it's all negative. So in 8 minutes, we're going to come back and we're going to read that. And for sake of time on my video, I'm going to pause it, okay? So I'm going to start my timer, and I'm going to pause my video, and I'll come back in about 7.5 minutes. Okay, we are back, and our test is complete in about 7 seconds. 6, 5, 4, 3, 2, 1. Okay, so 8 minutes later, we're going to read our test, and our patient, Skipper, is negative for all of those tests, those pathogens, diseases that I had mentioned, the anaplasmosis, the ehrlichiosis, the Lyme disease, and the heartworm disease. So our dog has detected negative. In this upper left-hand corner is the positive control, and we want to make sure that that actually does happen. If that didn't happen, then this test would be invalid, and that would mean that something went wrong with our test, and we would have to repeat it. So, so in fact, it worked, and our dog is negative. So that is the ELISA method test system. Very good system, has high sensitivity and specific. So it's a really good test method, and a very reliable one that a veterinarian office can offer in their clinic. Thank you. In this video, I will demonstrate the proper method for collection, preparation, and evaluation of an ear cytology sample in a dog named Rico. First, before I'm going to collect my sample, excuse me, I'm going to moisten a sterile swab with 0.9% saline as to not cause any cellular damage when I obtain my sample in the ear canal of my dog. So while I get the dog ready, I'm going to pause my video. Okay, so I had moistened a sterile swab with 0.9% saline, and now this is my dog Rico. We're going to pull his ear back. We're going to apply our swab into the ear canal and rub all the way around. Good boy. Okay, so while I prepare my video camera, I'm going to pause the video again. So I have obtained my sample from my dog Rico, and now I'm going to take my swab and I'm going to roll it onto my slide a few times as to get it all the way around in the swab. So I want everything to roll onto my slide. Okay, and I've done that now. This is off to the side. Now, if there was an excess of earwax, as this may interfere with the evaluation of the sample, I could gently warm the slide by passing the slide through a flame or heat from a hairdryer. But I have to be careful that not applying too much heat, because that can cause cellular damage. So I'm going to properly label my slide so I know which side is up and who the slide belongs to, and I'm going to allow this slide to air dry. So I will pause my video and I will come back when it's time to stain this slide. Okay, so I've let my slide air dry, and again, like I said, I don't need to heat fix the slide. The only time I would heat fix the slide if I was doing a gram stain or melting the earwax, as I just mentioned. So I'm going to proceed with staining my sample. I've labeled it with the patient's name and the date and what it was, or what it is. So I'm going to dip it into my fixative 5 times. to just dab off. Next is into my eosin stain 5 times. Kind of let it drip down. Next into my thiazine stain. Let it drip. Next into my rinse. Next, I'm going to run it under the water with the opposite side of the application of the specimen, because I don't want to let my specimen rub off. Now I'm just going to dab it on the paper towel, and I'm going to let this air dry. And while this is doing this, again, I'm going to pause my video. Now if I wanted to read this slide rapidly, I could put it in between Bibulous paper and we could read it right away, but I'm going to let it air dry. Okay, I'm back. So now I have put my slide under the scope, and I am looking for any abnormalities, inflammation, any microbes, bacteria, any fungus, any molluscicea, which are characteristically of the peanut shape. So, and that can cause chronic ear infections. So I'm right now I'm under low power with oil on under my scope, and so that means I am at 100 times magnification, total magnification, and I'm scanning for any abnormalities, and so far all I'm seeing are epithelial cells, nothing abnormal in this particular patient. Now if there was some kind of yeasty thing going on, or I'm sorry, some fungus going on, like molluscicea, we would see that on here, and it would look like a characteristic peanut shape, dark purple. I could see microbes on here, bacteria, and in that case, it tells a lot. We can, the doctor will then definitely know what to do and what kind of medication to give. But with Rico here, we have a normal ear canal, which is quite good. So that's it. Thank you. Hello. Today's video is on the handling of and safety precautions for samples taken from rabies suspects. If an animal is suspected of being rabid, the vet and the vet tech need to avoid contact with saliva. They should wear personally protective equipment such as double gloves, protective eyewear, and disposable outerwear. They need to avoid contact with the cerebral spinal fluid at all costs and always wash their hands well after coming in contact. If a human has come into contact with a rabid animal, a post-exposure rabies vaccination should be given. There is a prophylactic vaccines available for those individuals who are high at risk of exposure. If a vaccinated animal is suspected of coming in contact with a rabid animal, that suspected animal should be revaccinated promptly and observed for 90 days. If an unvaccinated animal is suspected of coming into contact with the rabid animal, they should be observed for 6 months and not be vaccinated. The only way to test, unfortunately, for a suspected animal for rabies is by post-mortem. The animal should be decapitated. The head is then double bagged and placed on dry ice, cautioned on the box, overnighted on delivery to the laboratory. Thank you. Hello, here we have Lola, a 4-year-old intact Pomeranian, and we're going to collect a vaginal sample to demonstrate the technique involved, as well as the cytology that we will see after preparing the slide for microscopic viewing. To collect the sample, we take a swab and we moisten it with sterile saline. That's important because we want it to be less traumatic on our little patient here. And if the patient is in proestrus or estrus, their vaginal canal will actually be a little thickened, so they will actually take it. I'm going to start off, before I stick the swab inside, I'm going to clean the vulva, so as you don't contaminate the outside with the inside. That's important. Okay, so I'm going to take my swab. Okay, this one. And I'm going to take my swab and I'm going to enter the vagina, and I'm going to go in and I'm going to stick it in, so I'm not just getting a labia sample. I don't want to do that. I want to get a nice, good sample. Okay, now Good job, Lola. I'm going to take my slide. I'm going to roll out the contents of what I got onto my slide. And then I'm going to prepare my slide as I would a cytology sample. So for now, I'm going to pause my video and continue in a few minutes. Okay, now that we obtained our vaginal sample from Lola and we prepared our slide for microscopic viewing, I'd like to go a brief overview of the canine estrus cycle. It is a continuous cycle with 4 different stages. Stage 1 is proestrus, and that lasts for about 9 days. Stage 2 is estrus, and that lasts for 9 days, and ovulation is on day 2. Stage 3 is diestrus, and that lasts for 50 to 60 days. Stage 4 is anestrus, and that lasts for about 5 months. Now, during each stage, there are changes in the vaginal epithelium cells that relate to the stage of the dog cycle. It is these changes in the epithelium cells, along with the clinical findings from the physical exam and the behavior as noted by the owner and hormonal testing, that we can gauge in which cycle the dog is in. Okay, now we're ready to view Lola's vaginal cytology under the microscope. And I'm going to start off looking at her slide, and I put it on the stage here, under 10, using my 10 times objective here, because I want to focus in on my cells, which I have. So then I'm going to grow up. I have a 50 on here, which I really love my 50 times objective, but for these purposes, I'll go up to 100. So that's 1,000 times total magnification. And as I'm scoping around here, I will discuss what I'm looking at. Now, there is a lot of bacteria on the slide, and there are some red cells, not a lot, but some. So what I'm looking for, there's 4 different types of cells that I'm looking for. The first one would be a parabasal, and that's a baby cell. It's kind of like small, round nucleus, and the cytoplasma to nuclear ratio is barely there. This type of cell is found in proestrus, diestrus, and anestrus. The second cell, which, by the way, I'm not finding any parabasals on this slide. The second cell is intermediate, and this comes in small and large. The cytoplasma to nuclear ratio is bigger. The small intermediate is still round but bigger, and the cytoplasma to nuclear ratio has increased. The large intermediate is larger with an irregular shape, as well as the cytoplasma to nuclear ratio increased. The nucleus is both small and large and is smaller than the parabasal nucleus. And we find that type of cell, the intermediate, small, and large, in the proestrus, diestrus, and anestrus. The third cell we see is called the superficial cell. And by the way, I am seeing a lot of intermediate cells in Lola's sample here, both small and large. And I can bring it up on my camera in a minute and show you what I am seeing. I'm just still scoping around, but all right, so where are we at? The third cell is a superficial cell, and this looks like a squamous cell that you would find in urine. A lot of cytoplasma with a small nucleus. The cytoplasma will fold over itself, can fold over itself, doesn't have to, but it can. And with the superficial cell, you'll find this in the estrus stage at greater than 90% of this type of cell in the estrus stage. I'm not seeing that in this slide. So Lola is not in estrus, but so far it's based on what I'm seeing, it looks like she's in the late proestrus stage, about to enter estrus. Okay, so the last cell I will see, or can see, is anuclear, and just as the word says, it's without a nucleus. And it's somewhat like a superficial, it's smaller and without a nucleus. And this cell is in the estrus stage as well. So if Lola was in estrus, I would predominantly see greater than 90% of superficial cells and some anuclear cells. But I'm not seeing that here. What I am seeing is small and large intermediate cells, a few red cells still, because Lola is leaking some, discharging some red discharge, and she has a swollen vulva. So let me see if I put my camera to my. All right, can you see my computer? On my camera here, on my microscope, this is one of the images I'm seeing here, which is a large intermediate cell. It has see all these little pink dots are bacteria. It has a large cytoplasm with a round nucleus. Now, it's not yet a superficial cell. It does not small and pygnotic like a superficial cell. So this is still a large intermediate cell with a bunch of bacteria all over it. So based on what I've seen on this slide and based on the physical exam of Lola discharging some red pink fluid from the vaginal area and her swollen vulva, I would say gauge her to be in late proestrus stage, about to enter estrus. And I hope I have demonstrated that technique well. Hello, today's video is going to be on performing a major and a minor cross match. Now, here I have 2 equine blood samples. One is from ACE. We have serum, which I already spun down for the sake of time, and we have EDTA blood, okay? And that is ACE, and he will be our recipient. Then I have the second horse, which is Jazz, and again, a serum and EDTA tube. And I spun those down for sake of time in my video. All right, so let's pretend ACE needs some blood, and Jazz is going to be, hopefully, our donor horse, and hopefully they will be a compatible product here, and we can proceed with transfusion. So the first step is I want to decant my plasma. I want to get rid of my plasma from my purple top, and I already pre-labeled some tubes, again, for the sake of time in my video. So I'm going to get rid of my plasma into another tube, okay? So I have going into this tube ACE's EDTA plasma. So I'm going to take all the plasma out, and I'm going to put it into here. Okay, so when that part is done, I'm just going to cap this off, and I'm going to move this over to the side, because I do not need that for any more of our procedure. Same with Jazz, our donor horse. I'm going to decant Jazz's plasma. And really, I can throw it away. I really do not need it for any part of the procedure, but I'm going to save it for now. Okay, cap it and put it off to the side. All right, so the next step is we are going to, according to the procedure, even though I have a serum separator in this tube, I'm going to decant off the serum, and because I have a serum separator, I don't need to use a pipette to take that off. I could just simply pour it into the tube. So ACE's serum here is going into the tube labeled ACE's serum. So I'm just going to pour it off like that. Cap this tube, put it off to the side. I don't need it. Jazz's tube for his serum, I'm going to pour off into a tube labeled Jazz's serum. Okay, and I'm going to get rid of this tube. So one thing about doing a cross match is it's very important to keep yourself organized. There's a lot of tubes involved, a lot of steps you have to do. So it's really, really important to not get distracted while you're doing this, to stay focused and really stay organized. All right, so the next step, and I have a procedure here that I'm going to follow because I don't want to pretend I know this at memory. No matter how many times I'll do this, I'll still look at the procedure. So the next step, we're gonna prepare the blood. We're gonna wash the red blood cells. Okay, so we're gonna turn this around. Okay, so this is Ace's side, and this is Jazz's side. Okay, so just gonna remix Ace's blood. Remember, I took the plasma off, so this is just red blood cells. But I want to wash them. I want to prepare them for the cross match and get rid of anything that may cause artifacts or false positives or anything like that. I want to wash my cells. So into this tube, I have Ace, which is my recipient. 2 drops of his red cells I'm going to put into this tube. So I mixed my EDTA blood. I'm going to take 2 drops. 1, 2. Okay. I do not need that pipette anymore. And into Jazz's tube labeled Jazz being the donor, 2 drops of red cells I need to place into here. And again, I want to mix this. Okay. 2 drops of Jazz, our donor. 1, 2. Okay, so now we are going to, let me put this over here. We are going to take our blood bank saline, which is a 0.9% sodium chloride, and we're going to fill this tube up because we are washing these red blood cells. Okay, same thing with this one. I'm going to fill it up. All right, now I'm going to cap these both. And I'm going to invert them a few times because I want to mix them up with the saline. I want to mix the blood with the saline. Okay, so here I have a centrifuge. And I need to spin it at 3,400 RPMs for 1 minute. Okay, so while that's going, let's talk about what I'm doing. I'm washing the cells again with the saline. When it comes out, I'm going to decant off the saline, and I'm going to repeat this process 3 times. And why I do that, I'm going to pause my video so that I save some time on my video. Okay, so and when the tube comes out, the cell button should be on the bottom. It's kind of fixed to the bottom of the tube, so simply I just pour it off into the sink, the saline, and resuspend my cells. Fill it up again with the saline, put the cap back on, invert it in my cells, fill it up with saline, and my cells, fill it up with saline, invert it a few times, and this is going to be wash number 2. So like I said, I'm going to pause my video. Why I'm doing this for the sake of time, and I will come back to this procedure. Okay, we're back and we have washed our cells 3 times. So now I'm going to make a 2% suspension of my cells. So I'm going to take a drop of my washed cells and put it into a tube labeled 2% suspension. And that was Ace. Now I'm going to do Jazz. Okay, then I want to make a 2%. So I'm going to add about that much. And I'm going to resuspend it by simply flicking it with my finger like that. Okay, so that is Ace. Add some blood bank saline to Jazz's. Same thing. Okay, so now I have my suspensions. I'm done with these cells. I'm going to move them to the back, and I'm going to perform my major and minor cross match. So into my major cross-match tube, I'm going to put 2 drops of ACE's serum, which is right here, into that tube. And why I'm dispensing serum to that tube, I'm also going to dispense the serum into ACE's control tube, okay? So we're testing for auto-agglutination in each animal, in each one of our, and we're going to utilize that as a control. So 2 drops of ACE's serum into our major cross-match tube. 1, 2. 2 drops of ACE's serum into ACE's control tube. 1, 2. Okay, now to our cell suspension, I'm going into our into our minor cross-match tube. I'm going to add 2 drops of ACE's cell suspension tube contents from that tube into the minor cross-match. Okay. 1, 2, and now into ACE's control tube, I'm going to add 2 drops of the cell suspension tubes. Okay, so ACE's control tube is complete. All right, so we're not quite done yet because now we got to get our donor cells and serum going. Okay, so into our major cross match, now I'm going to take my donor cell, which is Jazz's cell suspension. I'm going to add 2 drops of my donor cells. 1, 2. And now, since I got a pipette in that, I'm going to go to my donor control tube and I'm going to add 2 drops of suspension to that. 1, 2. Okay. And now I'm going to go to Jazz's serum and I'm going to add to my minor tube, I'm going to add 2 drops of Jazz's, or my donor, serum to my minor tube. 1, 2, as well as to my donor control tube. 1, 2. Alright, so at this point I'm going to mix everybody. My major cross match, which composes of my recipient's serum to my donor cells, my minor cross-match tube, which is my donor serum to my recipient cells, and then my 2 control tubes. Okay, so I have 4 tubes here. So what I'm going to do now is I'm going to incubate them at room temperature for 30 minutes, and I'm going to pause my video for sake of time. Okay, the video was paused, I had a timer going for 30 minutes, which is now stopped. Okay, so we have incubated at room temperature all 4 of our tubes, our 2 control tubes, and our major and minor cross-match. So now we are going to centrifuge again at 1 minute. No hemolysis, which we shouldn't see hemolysis. Resuspend the cells. I'm looking at it under a light source. And a nice suspension of cells. So what have we determined so far? Macroscopically, there's no agglutination and there's no hemolysis. Good thing. Good thing for our recipient. So we don't stop here. We will also look at it under the microscope to make sure microscopically there's not a minute amount of agglutination that we're just not picking up with the visual eye. So for sake of time, I'm only going to show you an example of one time, and let's go ahead and use our major cross-match tube. I'm going to put 1 drop of that onto a slide with a cover slip, and while I get my microscope set up, I'm going to pause my video. Okay, we're back, and I looked at it microscopically, and there's no agglutination, which is good. It confirms our macroscopic observation. And I'm going to show a close-up of, I have a nice camera on this microscope, and let me see if I can show you the camera version of what I'm seeing. Now there is a little bit of rouleau formation, not to be confused with agglutination. If your cells and your donor, if your donor and your recipient were agglutinated, you would see like grape-like clusters on there. But normal rouleau stack formation is normal. so that's good. So what have we determined? We just determined that Ace is hits a go ahead to go with our transaction or our transfusion with Jazz, our donor, for transfusion. Hello. Today we're going to do skin scraping on Skipper here. Skipper has some alopecia along with some crustiness on his left ear. So we're going to check him for external parasites such as Demodex or sarcoptic mange. He's been checked with the wood lamp for ringworm and that showed a negative result. But just to make sure, he does have a fungal culture going for that. We're going to start off this slide with a drop of mineral oil that I already put on my slide. And I'm going to take my scalpel blade and I'm going to dab both sides of my scalpel blade into the mineral oil on the slide. Now, as I'm scraping Skipper's ear, I'm going to avoid slicing him because I don't want to do that. I want to just scrape the infected area, and I'm going to go deep into the hair follicle as well, where the mites tend to be, and I'll probably pull up some blood as well. So as I am getting the camera ready to view that procedure, I'm going to pause it. Okay, so now I'm going to take my scalpel blade here and I want to scrape away at the infected area on both sides, getting into the hair follicle. So hopefully we can pick up some mites or parasites. Stay, good boy. And get some sort of idea what's going on with Skipper. Okay, so we got some material, and I'm going to now place that stuff that I've pulled off of Skipper's ear, and both sides I'm going to place onto my slide here like this. So it goes into the oil. Okay, now I'm going to take a cover slip and put that on, and then I'm going to view the material under my microscope. And as I do that, as I prepare my microscope, I'm going to pause the video. Alright, so here we are at the microscope, and I am going to scan my whole slide here on low power, which is 10 times using that objective, which is 100 times total magnification actually. So I want to scan my whole slide for any mites. Now, should I find something that is suspicious, I will go up to 40 times, which is actually 400 times total magnification. And then I can get a bigger view of what I'm seeing here. So far, I don't see any mites. Well, now here's something suspicious. So I actually will go up to my 40 and see what it is, but it is not a mite. But you get the idea of how this works. So I don't see any mites on this skin scraping, but I hope I have demonstrated a good technique for skin scraping. Thank you.